Today we will talk about Star tool. Our main focus is actually in the last class we trimmed our FASTQ file and got some result that was actually paired in file. So after trimming, what the output we have. In further, we will map all the reads to a reference genome. So give me a recap. In the last class we generated these file after trimming. Here we have paired file and also we have unpaired file. The paired means the reads that actually map and it has its mate or it has its pair. And after trimming, if the match or pair is removed, then it is actually goes into the unpaired file. So we mainly focus or we mainly analyze the paired file level. So trim paired the first queue and trim paired the first queue. This is for the R file. If we want to read something about that file, so here we are reading the first read. You can see the first one, the second one, and the third one. So we have a few amounts of read in this file, and what we will do, we will go one by one. That is first this one, then this one, then this one. We will go one by one from first, that is the beginning, to the end, and we will try to map each read with a reference genome. You know about reference genome, that is for human we usually use HG38 or HG19. For today we will focus on a reference genome that I downloaded from the gene code. This is the assembly, genome assembly, and this is the GT file. So now let's focus on the thing. So here you know about the book Hamlet written by William Shakespeare. So it has many pages. It's a complete book, and here you can see three most popular words or sentences from that book. The first one, to be or not to be. That is the question. Something is written in the state of Denmark. The lady to protest too much meeting. So here could be two situations. That is, we need to find out these three sentences. In the what situation come to the point, we know the pages, we know the page number where this situation actually located. And another situation we don't. We have no idea where this situation is in the book. So in that case, what we need to do, we need to read from the beginning to the end of the book and search for these three sentences in the book. And in this way we can find a match. That is, okay, so in the book, suppose page number 78, we found the match. So you can think about our genome as like a book and the read as like some sentences. So what we need to do, we need to find out the location or position where those read actually map with the genome. So this is the thing we try to do using aligner tool or in alignment. If we talk about the Star tool, Star actually uses its own algorithm. It's actually called a suffix array. The suffix array actually what it does, we have genome and it actually contains 3 billion bases. So if we want to search some read that's actually 100 to 150 bases, it's hard to find out that read in 3 billion bases. For that reason, we need to somehow present those 3 billion bases in a way that will be efficient to search. And in this way, the suffix array actually very efficient or very handy. So how it actually represent the sequences? Here I am just taking an example. Suppose banana. When we take banana, first thing we need to generate suffixes from banana. How we need to generate suffix? First, take the whole word banana. After that, remove the first letter and take other. That is banana. After that, go for remove this first one, then nana, then remove first one, anna, then na, then a, and last one. This actually represent, this sign represent the end of the letter. In programming, we represent the dollar sign. We use the dollar sign to represent the end of a letter or sentences. So these are the suffixes, suffix strings, and after that, usually these suffix strings are sorted in lexicographically or alphabetically. In that case, A comes first, and here is the position where actually A is situated or located, and then A anna, then anana, banana. These are actually lexicographical short. And after that, when we got this, and we can see another example with a DNA sequence. Here you can see this is the actual sequence, that is the ATTCATG. We are taking whole word or whole sequence at first, then remove the first one and take others, remove the first one and take others, first just like this, and we are just sorting lexicographically in here. After forming this suffix string, we need to ensure or we need to make our suffix tree that actually works like this. And this is the thing where we actually search. So how we search? Suppose I want to get banana. So how? So this is our root in here. From this root, we will search for the first one. That is a. Here we got A. Then we have two options. Here Na also. Here Na also. And we can go for one only. So anana, we got the anana in here. If we want to search for anna, we can also go for it. Anna. If we want to find out banana, it will be here. So it's something like work this, that is the tree is the read or is the suffix string in a tree like structure that has actually branch or leaf. If we think about this node, these are actually leaf, and it works like na, and here we take na, and if we wanted to find out the only na, then it actually goes here na, and it will be the in. So it's actually some thing easy to find out reads. I hope you understand a little bit of the organization of the genome, how actually STAR tool organize this. After that, we can go for our analysis. So today, what we will learn the indexing, that is the genome indexing with STAR. Then how to align or map paired end or single in read to the reference genome, and why we need the splice junction. And after that, we will do some alignment assessment and the interpretation. And these are the files what we got in the last lecture. We analyze this, and if we want to go for the single int, then this will be the file. STAR tool needs some resources to map or perform its operation. That is usually our reference genome lasts up to 3 GB. Then what annotation file, it could be 100 MB. For those mapping, we may need to RAM 32 GB and some disk space 30 GB. Okay, so why the splicing things important? We are talking about mRNA or RNA, and we have other aligner tools. That is, if we think about the BWA Bounty2, you may heard the name, and we are dealing with the STAR tool. So what's the difference is actually BWA and Bounty2 doesn't aware of splicing things, but STAR tool takes account the importance of splicing. So here in a DNA or in a gene sequence, what we have mainly actually exon and intron. And when we go for RNA, the intron sequences are spliced out and we get the exon only. After that, when we will, okay, suppose we have these read, these are the read, and when aligning these read, we the first portion or a partial portion of the read map to the exon 1 and a partial portion of the read map to exon 2. So we have a single read, but it is mapping to two different exons. Here actually the splicing awareness comes out. We saw here that is for exon 1, then intron 1. After splicing, these 10,000 bases or these 5,000 bases are actually removed from the RNA seq. When we try to align with the reference genome is actually the DNA. Reference genome actually DNA, and it contains the intron and intron sequences. But what our read, this is actually the RNA, and it doesn't contain any intron. But when we will map these this RNA that doesn't contain intron with a DNA that actually contain intron, so we have some difference, we have some space because of intron. So when it actually maps exon 1 and exon 2, but if it is not splicing aware, then it could match to misleading. That is, it will match exon 1, then it will match the other part with intron 1. So that will be actually misleading. So this is why actually we need splicing. Here, why actually we use STAR. STAR handles splicing. It uses suffix array that is efficient to store the genome and look into the genome. It has the accurate alignment. STAR usually produces most accurate alignment. So it is usually popular or mostly widely used for its accuracy. It can discover novel junctions, novel splicing junctions, and what the most unique things STAR has, it generate the gene count files. So when it outputs or generate outputs, it actually generate a file that counts genes. Okay, we will see it, the files. So first, what we need to do, we need to build our genome index. So for that, the command is actually used. That is, suppose this is the STAR command, and when we run the command, we have a few modes, and we need to index the genome, or we need to index the reference genome once, and we can reuse it again and again for indexing. And for indexing, what run mode we need, or what mode we need, that is the genome generate mode. And STAR tool supports the parallelization, and we can use any number of CPU threads in here. So suppose, how can you decide, that is, how many CPU cores you have? You can simply use a command nproc. It's actually in Linux. So nproc, this command returns how many CPU cores you have in your system. So here you can see 16, and you can when we define the CPU cores or threading, or when we decide this n part, we can just leave, that is we can leave some space in the code. Suppose we are choosing 12 or 8, something like this. That is, we usually don't using all the codes is a good idea. And here we need to choose the genome dir, where our indexing file will be stored. The genome FASTA file, that is the reference genome file path. Here is the gene annotation file path. And okay, so this actually is splicing junction. S for splicing, J for junction, D for database. And we usually need to choose this value by read length -1. We had read length 101, maybe you know 101, and this value will be 100. So after running the indexing, we will have some value like this. When we will run the command, we will get some notification in here that is started running. Then it's generating genome files, processing the annotation GTF. Okay, so here is the thing: starting to short suffix array. So this is the thing why Star actually index the genome in a suffix array. After that, it short the suffix array and save into the disk. Then it generate the index for that suffix array. Here it insert junction for the genome indices, and all the result it write into the disk, and thus we get our successful indexing. So I am not actually showing the indexing part in the lecture. It will take some time, that is 30 to 40 minutes. So here I am just showing what we get after the indexing. So here we use genome index as our genome dir path, and our all the generated things or all the generated indexes will be inside this directory. And here you can see the genome. This contains it's actually contains the genome sequences from our genome FASTA file that we gave. Then this is the SA, that it's actually the suffix array. SA means suffix array, and this is the index for this suffix array. And what file we actually saw HJDB, these are actually helps in the splicing junction, and it created from the GTF file we gave. And this file actually used for the mapping coordinate purposes, and you can ignore the other ones. The stat tool uses this one for its unique purposes. Okay, so here you can read this why this file actually use the genome, then SA index. These are the core alignment purposes, and HJDB list and all these tools are used for the splicing junction, and it actually contains the exon boundary. So after that we need to, so I actually did the thing in here. Here is the command we run for the stat tools. How it works, we need to specify the indexing path. So it actually in our current directory and genome index, and here is the file input. We are passing two files as it is paired in this one, first one, and this is the second one. If we have single in file, then we will pass only one, that is maybe this one, and we are specifying the threading as 8. And here this command is used to specify the folder path directory and what will is file actually start with. This is the prefix. This prefix will have all the files, and we want BAM file as our final output, and the BAM file should be sorted by coordinate. And if we don't specify this one, that is the quant mode, then we won't get the gene counts file. So these two options, if we get these two options, is actually for the less RAM option. That is, if we specify this one, that is the genome load, it will reduce our RAM usage, and we are limiting our sorting BAM file. Usually consumes huge amount of memory, and we can just limiting the RAM for this. That is how much RAM it can use. After that, we can see star run. Then it is loading genome. Then it started mapping. Then finished mapping, and now it is sorting the result, sorting BAM file, and it finished successfully. Now we can see the output, what it actually generated. This is the main point for today's class. So our result will be inside the mapping, and it will contain the prefix, this one. So we can go to our directory, and here is the mapping. What the output is actually generated. Here, this is the file that we will use for the further analysis. So this is the main output, you can say. And another main output is reads per gene. This is the gene count file. So what it does, what it contains, here it contains the gene name in symbol format. You can see the gene name in here. And for these first 4 rows, it contains n unmapped unmapped means read didn't map with the reference gene. So what read we had in our FASTQ file that actually doesn't map, and what is the count of that? This is the count. And some read actually map multiple location. Multiple location that actually means actually we have a read and we found that read in many locations instead of one. So when we get that one single read in many locations, that's actually called the multi-mapping. And in no feature, this actually means it actually mapped the read, but it couldn't assign any gene for that mapping. So when we don't get any gene, we get the count in this. And in ambiguous, so it has some this mapping has some ambiguity actually. And here, if we think about each gene count, here you can see the zero mainly contains zero. So we can go in here for this gene. So here you can see 4 columns. First one is the gene column that contains the gene ID or gene name. Then this is contains the count of both stranded. Okay, so the third column actually contains if the read map the forward strand. So how many times it map forward strand? One time. How many time it map the reverse strand? One time. And this is the count of. Here you can see another example, that is this gene. It map 4 times in the forward strand, 11 times in the reverse strand, and the count is 15. And it actually the unstranded. So first column is unstranded count, second one is forward stranded count, and third column is reverse stranded count. And modern tools actually, so when we use modern tools actually use the last column. When we will do the differential gene expression analysis in the fourth lecture, we will use the last column actually. So this count for each gene. And another important file is the log file in here you can see. So it actually contains how many input read we had in our first Q file. After that, how many uniquely mapped. This is the percentage. So how many map, how many read. So we had total 298 plus read, and among those 158 plus reads are mapped with the reference genome, and it actually 53.02%. And how much unmapped? So you can see in here the unmapped reads, and here the multi-mapping reads. So these are all actually contains the unmapped reads. Unmapped reads due to the it's the reason why it's actually unmapped. So unmapped too many mismatches, it actually zero. Then unmapped due to the too short length. So after trimming, we saw in the last class, after trimming we get some read that are very short length, and we remove some of them, and some may exist. And it often actually hard to find out a unique map for a short read. So these are the actually main thing: how much read unmapped, how much uniquely mapped, and how much mapped in multiple locations. Multiple locations means I just told you that is one gene, one read, and it is found in many positions or many locations. And if it is too many locations, and if it is found in too many locations, it's actually this one. And it is actually 0.70%. Now we can get an idea. So how many number is actually fine or how much isn't fine? So here, if the mapping, that is, if the count that is the uniquely mapped, if that is greater than 85%, then it will be good mapping. That is the excellent mapping. And for the multi-map, it need to be less than 5% to be excellent. And for the unmapped, it should be less than 10%. And annotated splices. Okay, so here we had another thing, the splicing junction. This section actually, and it has some percentages. So this should be greater than 95%, and the acceptable alignment, that is the acceptable range, should be 70 to greater than 70%. For the multi-mapping, it should be less than 10%. For the unmapped, it should be less than 15%. And the annotated splicing, there is a splicing junction mapping that should be actually greater than 85%. So if we come back in our genome, that is in here, we can see the uniquely mapped percentage is actually very low. So it has 53%. So it could be due to many reasons. That is, we choose the wrong reference genome file that actually doesn't align with these reads. It could be we are using AG38, but that read actually from the AG19. It could be actually. So here, if we get these uniquely mapped reads less than 70%, then it's a little problematic, and we need to find out the region. And for the multi-mapping, it should be less than 10%. So here we are getting 11.8%. So it could be a little problematic. We need to identify or find out the region why it's happening. And here for the unmapped reads, it actually okay. If we get less than 15% unmapped reads, that's fine. So what we actually needed to them. That is, ISTAR tool, it indexes its genome using a suffix array. That is, it stores or organizes the reference genome into a suffix array, and then it started its mapping the read from the FASTQ file, that is from the beginning to the end. And in that process, we got some uniquely mapped reads. We got some multi-mapped location. We got unmapped regions. And how much percentage we are getting for those regions, it's actually important to identify or to assess the quality of the mapping. So if we are getting less than the standard percentage or number, then we need to recheck what's actually happening or what could be the wrong. I hope you got some idea in today's class. So if you have any question, you can ask now. I will suggest to run the star command in your own machine, and it will be more beneficial for you, and you will find out the question or the problem where you are having the issue. Abdul Majid,
Yes.
you can actually finish today's lecture.
Okay, if no one getting any question, it's fine. Finish this session for today, and hope so you all understand very well. And if you are facing any problem while performing on your own dataset that you have chosen for your analysis, you can let us know in the group. Okay, so here I will stop the recording.